We will be talking about diseases of Bhindi. The botanical name is Abelmosus esculentus, family is Malvaceae. We will talk about yellow vein mosaic. The causal organism is the Bhindi yellow vein mosaic virus or Bhindi vein clearing virus. It has a good economic importance. It is very serious constraint in Bhindi production throughout the country. The Gemini virus is having spherical bipartite particles. It has a single-stranded DNA as its genome. The symptoms includes yellowing of the entire network of veins in the leaf plate, which is called as vein clearing. It is the characteristic symptom. In severe infections, the younger leaves turn yellow, they become reduced in size, and in the plant it is highly stunted. In a field, most of the plants may be diseased and the infection may start at any stage of plant growth. The infection restricts flowering and fruits are not formed. If they are formed, they are smaller in size, they are very hard and very rough. The loss in fruit yield ranges from 50 to 100% based on the disease incidence. In order to go for its management, we can go for tolerant varieties, which includes Parbhani Kranti, Usa Savani, Janardhan, Harita, Arka Anamika, and Arka Abhay. We can also go for removal and destruction of infected volunteer plants and collateral host in the vicinity of the field. We can go for erection of yellow sticky traps in the field to trap the white flies. Vector management for restricting spread by spraying systemic insecticides like monochlorpropose 1.5 ml per liter, dimethoate or methyl S dimeton 2 ml per liter. Here you can see the image of the disease. Both the images are of diseased leaf. Here you can see the next image of the diseased leaf. Both the images are of diseased leaf. We will talk about diseases of beans. Anthracnose. The causal organism is Colectotrichum lindemuthianum. The symptoms include all the above ground parts are affected at any stage of crop growth. However, the characteristic symptoms appear on the pods. On cotyledons, spots are sunken, dark brown or black with pink spores mass. Seedling infection results in collapse of seedling. Spots on leaves appear on lower side and are black. Later, these may also appear on upper surface. When the infection is severe, the affected plant withers. Black, sunken, circular spots of varying size appears on pods with bright red, yellow, or orange margins. The center of these spots later turns gray or pink due to sporulation of pathogen. The border of these spots appear raised. Here you can see these spots on the bean. The pods are nearly destroyed. Mycelium or conidia on seeds and collateral host, including cowpea, mung bean, black gram, are the primary survival methods. For management, we use healthy seeds for planting. We periodically harvest all mature pods and sort and destroy infected ones. Seed treatment with carbendazimine 2 gram per kg seeds. We protect the crop by spraying binomial or carbendazim 0.1% or Zineb or Mancozeb 0.25% at 10 to 15 days interval. Rust. On beans, it is caused by Euromyces fascioli typica. On cowpea and other, it is caused by Euromyces fascioli vegani. On dry bean, it is caused by Euromyces appendiculatus. Autoecious macrocyclic rust, that is, it is produced on all the stages on bean plant. Symptoms include pustules. They mostly appear on leaves, though they are also found on petiole and stem of some host. The rust pustules appears on either side of leaves, but more common on lower surface. The uridosauri are minute, roundish, slightly raised, and reddish brown in color. These contain rust spores and appear in groups. Later in the season, with the formation of teleospores, the sori turn dark brown or black. Diseased leaves may wither or fall off under severe infection. Here we can see the diseased leaves. It is the upper side of dry bean leaf with rust. You can note the yellow halos. Survival and spread. Primarily, it survives through teleospores in cooler regions and on collateral host. Secondary, it is spread through wind-borne uridospores. For management, early sowing is recommended to avoid severe infection of plants in the field. We can also protect the crop with Mancozeb or Zineb 0.25% or wettable sulfur 0.3%. Yellow mosaic or bean yellow mosaic virus or mean bone yellow mosaic virus or fasciolus virus 2. It is a Gemini virus having single-stranded DNA. Symptoms include bright yellow patches which appear on leaves. Yellow areas alternate with dark green area of leaf plate. Younger leaves show more severe mottling and chlorosis. Leaves completely turn yellow and gradually become necrotic. Plants are stunted and flowers and pod is said to reduced. Pod formation is reduced and if produced, they are deformed having shriveled and underside seeds and may exhibit molting when green. Here we can see the diseased parts of the plant. Primary spread and survival virin in collateral hosts like French bean, soybean, red gram, xanthium, strumerum, Eclipta alba, proton species, black gram, green gram, etc. Secondary spread and survival virin is transmitted by whitefly, Bemisia tabaci. The management, we can remove the collateral host and destroy from the vicinity of the field. We can use resistant varieties and we can manage the vectors by erecting yellow sticky traps and spraying systemic insecticides like methyl dimetone or dimethoate 2 ml per liter. Common bean mosaic virus or green mosaic or bean common mosaic virus. Virus with long flexus rods, it has single-stranded RNA. Symptoms. It affects only beans, Felis vulgaris and other Fasciolus species. Leaves show mosaic pattern, that is light green areas alternate with dark green areas. Diseased leaves, they become rough and show blisters on the leaf lamina. Leaf size, petiole length and plant height is reduced. Leaves become curl and they curl downwards. Diseased plants produce fewer pods which are smaller in size. The seed becomes smaller, malformed and is aborted. Alternaria leaf blight. The causal organism is Alternaria jasmine and A. Alternaria alternate. Symptoms: in the leaves, dark brown spots they appear. On fumed conditions, the spots enlarge, covering large areas, causing brightening of leaves. Concentric signs can be seen. The disease also affects stems, petiole, and flowers. The mode of spread and survival. The disease spreads through wind-borne conidia. Epidemiology. The disease attacks Jathi Malli, J. grandiflorum, and Mullai, J. auriculatum. The disease is severe during winter months, October to December. In certain areas, the disease is noticed even up to February. For management, collection and removal of fallen leaves can be done. We can also spray with copper oxychloride 0.25% or mancozeb 0.25%. Here you can see the diseased leaves. Collar rot and root rot. The causal organism is Sclerotium rolfesi. Symptoms: plants at all stages are infected. First, the older leaves they become yellow, followed by younger leaves, and finally death of the plant. In the root, black discoloration can be seen. On the infected tissues, the stem surface white strands of mycelia and mustard-like sclerotia are seen. Management: we can go for soil drenching with copper oxychloride 0.25%. We can also go for heavy application of FIM with Trichoderma viride. Here we can see the disease spreading into the plant. Pillar D, phytoplasma. Symptoms includes the leaves become small, malformed, and bushy. In the place of flower, green leaf-like malformed flowers are formed. The mode of spread. The disease is transmitted by grafting and whitefly, Dilurodes circilidae. Management. Selection of cutting from healthy plants is one way. Spraying insecticide to control the vector is another method. Survival and spread. Primary is oospore in infected plant debris and soil. Secondary, sporangia and zoospores by irrigation water and rain splashes. Favorable conditions are low-lying field with poor drainage, high soil moisture, and frequent rains. Management, selection of healthy cuttings for new plantings, field sanitation by collection and destruction of infected vines and debris, providing with non-host crops like rice or maize or sugarcane. Changing basis of infected vines with Bordeaux mixture 1%. Spraying metalaxyl plus mancozeb, Rodomil MZ72 0.2% on infected and surrounding healthy vines. Biocontrol with neem cake plus Trichoderma viridi. Sclerotium, foot rot and wilt. Causal organism Sclerotium rholtesi. Symptoms are plants are susceptible at any stage of growth. Collar region is usually infected. White mycelial growth over the infected area of stem on which small mustard seed like Sclerotia upper, spreading of infected bark, vine withers and totally dries off. Survival and spread. Primary are sclerotial bodies in infected plant debris or soil. Secondary, autonomous growth of the fungus or water dispersed sclerotial bodies. Favorable conditions: if there is high soil moisture and high soil temperature, or if the relative humidity is high and the weather is warm. Management. Selection of healthy cuttings for new plantings. Field sanitation by collection and destruction of infected vines and debris. Providing good drainage. Drenching bases of infected vines with Bordeaux mixture 1%. Spraying carbendazim 1% on infected and surrounding healthy vines. Biocontrol with neem cake and Trichoderma viridi. Anthracnose or leaf spots or marginal blight. Causal organism, Polytotrichum capsicum. Symptoms: only aerial parts of the plants are attacked. Light to dark brown irregular spots surrounded by diffuse chlorotic halo develop on leaves. Spots become sunken, enlarged, and colse. Marginal leaf tissue becomes black, necrotic, and gradually spreads towards the center. Circular black lesions may girdle the stem, culminating in death of the vine. Here we can see the condition of the plant. Survival and spread. Primary is dormant mycelium in infected plant debris. Secondary, rain-splashed conidia. For management, field sanitation can be done by collection and destruction of infected leaves. We can also spray Bordeaux mixture 1% or copper oxychloride 0.3% or carbendazim 0.1% at 15 days interval. Diseases of eggplant. Brinjal. Botanical name Solanium melanogyna. Lamb. Family Solanaceae. Little leaf caused by phytoplasma. Infection is initially observed in one branch and later the entire plant shows symptoms. Reduction in size of newly formed leaves. Reduction in petiole length, making the leaves appear to be sticking to the stem. Affected plants have narrow, soft, smooth, and yellow, shorter leaves. Affected plants have narrow, soft, smooth, and yellow, shorter leaves. Internodes of the stem are also shortened. Axillary buds get enlarged, but their petioles and leaves also remain shortened, giving the plant a bushy appearance. Mostly there is no flowering, but flowers are formed, they remain green. Here we can see the issue with the plant. Survival and spread. Primary, the pathogen survival in collateral weed host like Datura fastuosa, Datura semonimum, Athranthus roseus, Argemone mexicana, and transmitted to eggplant by Jessets. Secondary, leaf hoppers, Ishimonas pistis. Management. Tolerant variety, we can grow them, which includes Pusa purple round, Pusa purple cluster, and Arca shield. Destruction of affected plants. Eradication of solanaceous weed hosts. We can also go for spraying methyl dimetone 2 ml per liter or dimethoate 2 ml per liter. Bacterial wilt. Causal organism. Ralstonia solanacerium. Symptoms. Sudden wilting and death of infected plants in the characteristic symptoms. The petioles of older leaves droop down and the leaves show epinasty symptoms accompanied by yellowing and stunting of whole plant. Typical browning of vascular tissue of roots and stems can be seen. From cross-section of infected plants, whitish bacterial ooze comes out. Survival and spread. Primary, the bacterium cells is both seed and soil borne in nature and overwinters in infected plants, parts in soil invite host plants and weeds. Secondary, bacterial cells spread through irrigation, water, or infested soils and agricultural implements. Favorable conditions, relatively high soil moisture and soil moderate temperature, favors the disease development. Continuous cultivation of solanaceous crop in the same field. Management. Growing resistant varieties like plant samrat, Arka nidhi, Arka skashva, Arka nilkant, Surya, and BB1, 44, and 49. Crop rotation with non-solanaceous host, green manuring with Bavaria species, biofumigation, soil solarization with transparent polyethylene sheet, 125 micrometer thick, for 8 to 10 weeks during March to June in nurseries. Biological control with Pseudomonas fluorescens, Pseudomonas tepsii, Bacillus species, and Erwinia species. Phomopsis fruit rot or blight. Causal organism Phomopsis vexans. Economic importance, the disease is severe in tropical and subtropical areas of the world. In India, it was reported in 1935 in Gujarat. The pathogen attacks foliage and fruits, but the latter phase is more destructive. Symptoms. The plants are attacked at all stages of growth, producing damping of symptoms in nurseries and collar rot on young plants. On leaves, circular to irregular, clearly defined grayish ground spots having light center appears. The disease leaves become yellowish in color and may drop off. Several black exindia can be seen in older spots. The lesions on stems are dark brown, round to oval, and have grayish center where pycnidia develop. At the base of the stem, the fungus causes characteristic constructions leading to canker development and toppling of plant. On fruits, small pale sunken spots showing concentric rings appear, which on enlargement cover entire fruit surface. These spots become watery, leading to soft rot phase of the disease. A large number of dot-like pycnidia also develop on such spots. An infection of fruit through calyx leads to development of dry rot and fruits appear black and mummified. Survival and spread. Primary. Pathogen is seed-borne and also survives in plant debris as mycelium and Pycnidia. Secondary, Conidia disperse through rain splashes, irrigation water, agricultural tools, and insects. Favorable condition, high relative humidity coupled with high temperature favors disease development. Maximum disease development takes place at about 26 degrees Celsius under wet weather conditions. Management. Procuring seeds from healthy fruits followed by seed treatment with thiophanate methyl at 1 gram per kg seed or hot water treatment of seed at 550 degrees Celsius for 30 minutes. Practicing crop rotation and summer plowing helps in reducing initial inoculum. Removal and destruction of diseased crop debris. We can also spray twice with thiophanate methyl or carbendazim at 0.1% at 20 days interval. We can also go for growing resistant varieties like Florida Beauty, Solanum Gillo, Solanum macrocarpa, and Solanum integrifolium. These are wild resistant